Okay, so hello everyone and welcome to our webinar today. My name is Anne McLean. I'm a senior application at Magritek. Before I hand it over to Kurt, our presenter today, just a few housekeeping items. So this webinar is being recorded and the recording will be sent in a follow-up email after the webinar for you to view on demand. During the webinar, if you have any question, please feel free to ask the question through the question box of the GoToWebinar here. At the end of the presentation, we will have a live Q&A session where we will try to get through as many questions as we can. And for those questions that we can get to, we will be sure to follow up with you through email. And with that, it's my honor today to introduce Kurt Zinalotti, our application scientist, and he will be taking us through the diffusion NMR experiment on our Benchtop NMR spectrometer.
Thank you very much for the kind introduction, Anne. And like she said, today we're going to be discussing Benchtop NMR diffusion measurements and how we measure things from mobility all the way up to structure property analysis using this particular instrument. So to outline, what we're going to do is we're going to showcase the use of NMR diffusometry to analyze and characterize complex systems in a variety of applications. These applications include transport properties for battery electrolytes, for example, utilizing multinuclear NMR diffusometry. We have polymer analysis that studies polymer blends and molecular weight analysis. We're going to look into some mixture analysis and using NMR diffusometry as a deconvolution tool to deconvolute overlapping signals. And last but not least, we're going to study some structure-property relationships using restricted diffusion measurements as a way to characterize porous materials. So overall, NMR is a very powerful technique that offers both non-destructive and non-invasive information for detailed structure determination, but it also utilizes and gives very valid information regarding internal dynamics of a system. So to start, the Spinsolve has been on the market for over 10 years now, and therefore we have 10 years of constant improvement, as we now have 3 instruments, all of which can be utilized for these diffusion measurements, and they range in different magnetic field strengths from 80 all the way up to, or from 60 all the way up to 90 megahertz. We have a wide range of add-ons as well. 2 of the add-ons that we're going to be discussing today is the Multi-X add-on, which allows for fully automated switching between nuclei to fully characterize complex mixtures, as well as diffusion gradients, which are needed to perform these experiments that go up to about 1 Tesla per meter in strength. So to start, what is NMR diffusometry? Well, NMR diffusometry is simply the study of molecular motion. And the NMR is really the only widely available technique that provides these self-diffusion coefficients. And we'll get into a minute of what those self-diffusion coefficients actually are. So what NMR does is it utilizes spatially varying magnetic fields to label these spin ensemble positions throughout the sample, and therefore are able to track their diffusion coefficients. And in turn, why do we utilize NMR for these types of experiments? Well, like everyone in this audience knows, NMR is a non-destructive and non-invasive technique. It offers superior chemical selectivity as through chemical shifts and through different nuclei that are able to be studied, you're able to fully characterize complex mixtures. And in turn, characterize things like just lithium and a lithium electrolyte without having to characterize the entire mixture by itself. So what are we measuring with these measurements? So with these measurements, we're actually studying random Brownian motion, in which random Brownian motion is really just random fluctuations of a molecule's position in time. So the diffusion distance here is represented as the final position of this red dot. As it takes a random walk, it ends up at its end of its course. And that distance is defined as its final position minus its initial position. We know the time it takes for this diffusion to occur, as that's represented in our experiments as this big delta coefficient. And in turn, with those 2 parameters, we're able to calculate a diffusion coefficient, which is typically represented as this large D throughout this presentation. So from that, we can use diffusive length and determine that through a root mean squared displacement equation, in which if you know the diffusion coefficient and you know the time you allow for the diffusion to happen, that large delta coefficient, you can probe diffusion length scales using NMR that range from hundreds of nanometers all the way up to tens of microns, which fits in a nice range between some other widely used techniques for diffusion measurements. So what are the basic theory behind these measurements? Let's simply look at one of the most basic diffusion edited pulse sequences, which is this pulse gradient simulated echo experiment, or PGSTE for short. Here I've drawn out a simple schematic of the RF magnetic pulses with the gradient pulses, in which I've labeled 4 different time points, T1, T2, T3, and T4, in which we have 3 parameters that we really care about, or that are widely used to edit this pulse sequence in real time. So we have a gradient strength, which we can vary all the way up to 1 Tesla per meter. That's represented as the G coefficient. We have a time in which that gradient pulse is on, which is represented as our small delta, and our big delta, and between those 2 gradient pulses is the time we're allowing for diffusion, or our diffusion time. Typically, that time scale is on the order of milliseconds all the way up to seconds, depending on what nuclei and what molecules you're looking at. So here I have an NMR tube that we're labeling spins in 4 different positions, being a red, yellow, purple, and green. And we're going to see how those spins evolve over the course of this basic pulse sequence. So at T1, we have the 90-degree pulse, which introduces the spins into the transverse plane. Here, the spins persist within the transverse plane, and we then apply a gradient pulse at a particular length, which is determined by that small delta coefficient. And what that gradient pulse does is it simply encodes the spin position by spatially varying the magnetic field across the sample. So now the spins experience a different precession frequency while the gradient pulse is on, which is theoretically encoding their position. We then turn the gradient pulse off, and we allow for diffusion to occur during that big delta or diffusion time. As diffusion occurs, spins can move throughout the sample, swap positions with one another during that T3 time point. And then at T4, what we're doing is we're applying the same gradient strength, but this time we're decoding the spin's positions. So the easiest way to think about this is if no diffusion occurred at T4, the spins would all persist within the transverse plane similar to T1. But if diffusion occurred, the decoding pulse cannot simply undo the encoding pulse, and now we have our signal, or our total available signal is now attenuated. So if we compare what the spin total vector looks like, at T1, we have our total gradient strength, or our total spin magnetization within the transverse plane. And if diffusion occurred, we can have our signal attenuated as in T4, and now our total available signal is decreased. So when we FT after data collection, this is what we could typically see. So we can vary the gradient strength, and if we vary the gradient strength, what happens within our FT spectra is we see signals start to become attenuated, and we can then integrate these signals as a function of the gradient strength to pull out diffusion coefficients. So what we do is we can plot the signal intensity as a function of the gradient strength, and then fit the individual data points to this equation here. This is the Spinsolve-Tanner equation, in which here we're then pulling out the slope, which is our diffusion coefficient. One thing to note here is all of these variables from the Spinsolve-Tanner equation are all controlled through the experiment or through the spectrometer you're using. And in turn, the only variable that is pulled out is from the slope, which is that diffusion coefficient we care about. So this may seem like a lot, but here at Magritek, we hope to simplify the accessibility of this experiment for any researcher that wishes to use it. And what we've attempted to do by that is actually here through this screenshot of the software interface when running these types of experiments. Here we have individual slices of the experiment in which we're increasing the gradient strength, in which we can simply integrate the peaks, and the spectrum or the software is going to plot that integration as a function of the gradient strength. That is, the software then fits these points to the Spinsolve-Tanner equation, and the diffusion coefficient is automatically reported to the user. Here, we recognize that many benchtop users are not your traditional NMR spectroscopists that would use this technique. So hopefully, we've introduced a straightforward way of measuring these diffusion coefficients, which is a very simple method setup that is complementary to your T1 or T2 relaxation measurements on this particular instrument. And hopefully, we've easily or made the data processing somewhat easy for you to readily process the data as you wish. So what we can do next is we can directly go into a live demonstration of our instrument to show you directly how that is obtained. So I will actually pull it up right now. And what we see, this is our Spinsolve instrument that we're going to be using throughout the live demonstration portions of this talk. It's right down the hallway from us. It's our 80 megahertz instrument that allows us to analyze complex mixtures through a wide variety of nuclei applicable on it. And what I've done is I've already inserted a sample with the auto sampler so we can go ahead and measure something directly. So I'll go into our proton folder and I will scroll all the way over to our PGS-TE pulse sequence, in which we can obtain a diffusion coefficient for this 200 millimolar paracetamol sample which is actually dissolved in protonated ethanol. So I'm going to go ahead and vary some of these parameters that are already optimized to fit our needs for this particular demonstration. I'm going to lower the number of scans. We have full control still over our acquisition parameters, including repetition time and acquisition time. We then have full control over our little delta, which is once again how long the gradient is active for. Our big delta is the amount of time we're waiting for diffusion to occur, and our max gradient strength we'll just set as 100% of what the instrument can give us. Here, we're just changing the number of points or number of data collection steps that we're taking, and we'll go ahead and hit start. So one thing to note here is changing the attenuation level is easily done by varying either the max gradient strength, how long the pulse is active for, so that's little delta component, or how long you're allowing for diffusion to occur through the big delta. What we'll do here is we'll look at a single spectrum being the first spectrum that is collected. This is with very little gradient strength, and therefore the 1D spectrum here is going to be representative of your simple 1D proton protocol. If we look here, we have the signals for ethanol, and once we start zooming in, we start to see the smaller signals that correspond to paracetamol with the carbon satellites of ethanol once again. We can then integrate the spectrum. If we click on that integration button, we can then click and drag over the signals we wish to integrate. So I'm going to integrate the one paracetamol signal here, which is going to be represented as the red data point. I will then start to integrate some of the ethanol signals, which are going to be represented by the green and blue data points. And you see as soon as they're integrated, they're actually starting to be plot here on the right as a function of all the parameters we care about, besides all the parameters we care about. One thing to note here is the fit is linear because we're plotting the logarithmic scale of the integrations. So besides showing just a single spectrum slice, we can show the entire stack spectra, in which if we click on the stack protocol, we're now looking at the spectra as a function of the gradient strength. So as the stack goes up from green to blue, the gradient strength used to acquire this data is actually increased, which is why you start to see the signal be attenuated as a function of that gradient strength, which is shown over here again to the plot on the right. So now that we have these individual data points, we can go ahead and fit the data points to the Spinsolve-Tanner equation through this button up here at the top. We see that they're fit to a straight line. And from that value, we can open up this integration tab to actually pull out their diffusion coefficients. So we note here that we have two different diffusing species. Again, the red represents the paracetamol, which is a bigger molecule than the ethanol, which is why it has a slower diffusion coefficient at 4.5 times 10 to the minus 10 meters squared per second, versus the two signals in green and blue, which correspond to ethanol, which have the exact same diffusion coefficient because they're part of the same molecule. So as this data is finishing collecting, a few other things we can do. We can pull the data off directly into an Excel file. Here, the data that's pulled off in an Excel file is the XY coordinates for this plot we're showing over here to the right. And in turn, we can also pull the data off as a PDF once it is finished collecting. So as this finishes up in the next 3 seconds, we'll actually pull the data off as a PDF just to show you what that looks like and how it's represented. So I'll click on the PDF button, and now we're going to generate a PDF readout of the particular sample we just measured. We have all the parameters that went into acquiring the data as well as the integrations. We have the stack plot that we were just looking at. And then down here on the next page, we have the integration table or the spectrum that is then fit to pull out the diffusion coefficients. One thing to note here is we have our fitting equations right here with our diffusion coefficients labeled in the bottom left. So what we'll do next is I'll go ahead and prep the instrument for collection for another sample that will start while we're going through the rest of this presentation. Sadly, this sample is going to take a little bit longer to actually acquire in comparison to that 200 millimolar paracetamol sample, just because it's a little bit more complex. So I'm going to start the data acquisition while we go through the rest of the slides. And once this sample is finished acquiring, we'll come back to it to analyze the data just to see what we see. So I've simply interacted with the auto sampler just to change the sample. I've ejected the paracetamol sample, and I'm going to go ahead and load the next sample we care about. And once that sample is loaded, I'll go ahead and put in the parameters that have been optimized for this acquisition and start the acquisition while we go back to the slideshow. So the sample has been loaded. Now we'll go back into the Proton folder, and we'll go ahead and start the acquisition for this particular sample. The sample is named according to what our naming convention is for this particular sample. Later in the talk, we'll discuss what this particular sample actually is and what we're measuring with this acquisition. But for right now, we'll jump directly back in to the slideshow. So now that we are back in the slideshow, what we can do next is go to the next slide, which is going over that data collection once again. We're basically determining paracetamol and ethanol, in which, again, the diffusion coefficients are corresponding to the size of the molecule. Here is paracetamol will diffuse slower than ethanol. Another common way to represent these particular coefficients is within a DOSY plot. So what a DOSY plot is a diffusion ordered spectroscopy plot. So here we provide an example of a mixture of porcine and paracetamol. One thing to note here, to the left, the blue is the paracetamol, the green is the porcine. And what we see is we have some unique signals that correspond to each individual component, but in the red, once they are mixed together, some of these signals overlap quite heavily. So this mixture can be run simply due to the chemical selectivity through the chemical shifts, but we can determine the diffusion coefficients of all species here, including the solvent, as is plotted down here to the bottom. So if we integrate the unique signals here in green, red, and blue, we can plot them to determine the diffusion coefficients of those species. But to take this experiment further, it can also be plotted as a diffusion ordered plot, simply by using the DOSY toolbox presented by this Manchester group down here in the bottom. So what this does is this plots the diffusion coefficient on the y-axis, in which the x-axis corresponds to the chemical shift. Here the correlations are set to be similar to that of a 2D plot like COSY or HMBC, but now you can easily track the diffusion coefficients as they vary with the chemical shift of the mixture. And again, this plotting process is quite easy. It's just taking the data folder into the DOSY toolbox, and the DOSY toolbox does everything else and produces the spectrum for you. So what we're going to show next is the use of diffusometry through many different applications. So here again are the applications that were selected to talk about during this talk. We're going to talk about transport properties in battery electrolytes. We're going to go through some polymer analysis and how NMR is a very powerful tool for that particular purpose. We'll then go into some mixture analysis and how we're using diffusometry to basically deconvolute overlapping signals. And then we're going to get lastly into some structure property relationships and how those are determined through restricted diffusion analysis. So to start, we're going to look at transport properties in battery electrolytes. But to make this possible, we're going to utilize the chemical selectivity provided by the Spinsolve multi-X. So the Spinsolve multi-X is our answer to allow you to study multiple X nuclei on a single instrument. Note there are no manual tuning required of the instrument. In fact, we have additional pre-calibrated protocols for every additional nuclei, including multiple heteronuclear correlation experiments. So you can correlate things like phosphorus back to proton or fluorine back to proton if you wish. Again, we also have multiple decoupling experiments. So now you can decouple your things like phosphorus from proton or proton from fluorine, for example. We have fully calibrated methods that allow for unattenuated switching between nuclei, so you don't lose any performance. There is no manual tuning or matching of the probe that needs to be done. And this allows for the complex analysis of many mixtures, or the complete analysis of many mixtures. So if we go into the application here, we're going to study the transport properties of battery electrolytes, in which what we're looking at here is lithium PF6 and DMSO. So lithium PF6 is a commonly used additive for many electrolyte solutions, and that's typically done to improve the conductivity of many liquid and solid state batteries. Here, the lithium PF6 is at a concentration of about 300 millimolar, in which what we're doing is we're going to track the diffusion behavior of the lithium, the PF6 anions, and the DMSO through the NMR diffusometry experiments to basically determine the complete transport properties of the system. So here I show the 1D analyses obtained from the multi-X instruments of this lithium PF6 and DMSO, in which all the analyses were done in about 7 minutes. So at 300 millimolar concentration, the proton spectrum shows the DMSO signal. The lithium spectrum corresponds to the lithium ion from lithium PF6. And what's very interesting here is in the fluorine and the 31P spectrum, we see a few different species. We see the PF6, which is corresponding as a counter ion to the lithium. We also see a PF5 and an HF signal occur in the fluorine, just due to the presence of water within our DMSO. That's going to hydrolyze the PF6 into both PF5 and HF. But using fluorine NMR, we can actually calculate the concentration of those particular species if we wish. And we also see the same thing within the 30 and 31P spectrum, which takes about 5 minutes to obtain. So In practice, NMR diffusometry measurements can be done on any nucleus you're able to obtain a 1D spectrum for in a reasonable amount of time. So these diffusion pulse sequences, like PGSTE and PGSE, are applicable for any nuclei. And again, here's what we show with this individual lithium PF6 sample, in which here we're plotting each individual nuclei as a function of the gradient strength, in which we can then track again the attenuation as a function of that gradient strength to produce diffusion coefficients. So if we were to integrate any of these signals, we can then plot that and pull out diffusion coefficients, which are represented here within this plot as a function of the diffusion coefficients versus the concentration of lithium PF6 within the sample. So what we see here is using proton, carbon, fluorine, and phosphorus NMR, we can fully obtain these complex mixtures and the diffusion properties of any species within them. Here, the error bars are representative of repetitive measurements. About a triplicate study was used to determine the error bars of each individual measurement. And both the PF6 diffusion from phosphorus and fluorine match quite well with one another as presented from the green and blue dots. One thing that's important here is we see that as the concentration of these individual species increase, the diffusion coefficient decreases. This is quite important in this particular application, as the diffusion of these species is linked directly back to the conductivity of these materials. So battery applications, you really wish to crank the conductivity as high as you can, and to do that, you really need to optimize the diffusion of certain species to hopefully lead for that better performance by increasing conductivity. So by hopefully optimizing the diffusion coefficients for lithium, say in a lithium-ion battery, you hopefully get a better conductivity, which in turn leads to a better performance of the system. So besides simple transport property measurements and battery electrolytes, NMR is widely used for polymer analysis. So the NMR polymer analysis really encompasses a wide range of applications, and it really starts as simply as just structure verification or just confirmation that you have an initiator and monomers present for the reaction. So this can be viewed through this individual spectrum of 4-vinyl chloride, in which the black trace is the 4-vinyl chloride 1D spectrum of a freshly prepared sample. We see that everything is nice and sharp, indicating that there really isn't a presence of the polymer. But after storage of this sample over a couple weeks in very poor storage conditions, we not only see that there starts to be new signals that arise corresponding to the polymerization of this particular monomer, but also the line width of these individual signals also broaden, indicating a higher viscosity for the sample through the polymerization process of this particular monomer. Besides these very easy experiments, polymerization or polymers can also be used in NMR to study conversion or degree of polymerization. So here I show signals of a PCl polymer in which we can easily track the percentage of the monomer shown in red to the percentage of the polymer shown in the yellow here. And we can plot that as a function of the gravimetric content of these two species, and we see that they plot linearly. Notice here that as polymerization occurs, you can see a decrease in the monomer concentration as that decreases in intensity. To determine degree of polymerization through this, it's really quite easy through a simple equation. If you know the amount of protons or fluorines that correspond to that particular repeating unit versus an in-group signal, and you have the presence of the in-group signal within your 1D analysis, simple integrations can be used to determine degree of polymerization. So here to the left, we show a 1D proton in which we have signals that correspond to both the in-group and the polymer. Integrating those can be easily used to determine degree of polymerization. From that, if you know the molecular weight of the repeating unit, you can determine number average molecular weight quite easy. Note this is also easily done through fluorine NMR as long as the in-group is separated from the repeating unit itself. So this technique works quite well when you have those two unique signals. But once you start seeing larger polymers, the repeating unit may dominate the spectrum while the in-group may be hard to analyze. Or in times the in-group may be the same molecular unit as the repeating unit, and in turn might overlap quite a bit. So there are ways to get around this. One way being an alternative method to analyze and determine molecular weight, and that's typically done through NMR diffusometry. So here we've used the PGSTE method on our benchtop NMRs for molecular weight determination. One thing to note here is all polymers can be characterized with this technique. If there are no signals available for the in-group, this technique really doesn't use the in-group signals to characterize anything. So therefore, we'll use NMR diffusometry to determine molecular weight. There are no solvent restrictions. Traditionally speaking, if you have a solvent that is able to dissolve a polymer into solution, that can be used on NMR to measure your molecular weight. One thing to note here is the diffusion coefficient that is produced or obtained from this study is the absolute diffusion coefficient. What I mean by that is this diffusion coefficient will match what you get with high field instruments, as the diffusion coefficient does not vary with magnetic field strength. One thing to note here is you can really measure many different molecular weights here, as our Spinsolve traditionally has strong gradients that allow you to even measure high molecular weight polymers that diffuse quite slowly. So how is this done? So this is done through basically the Stokes-Einstein equation, which we plot here on the left. One thing to note here is the Stokes-Einstein equation takes two important parameters, one being the solvent viscosity, the other one being the hydrodynamic radius, in which we can correlate the hydrodynamic radius back to the size of the polymer, as long as the polymer is not interacting with one another. And what I mean by that is sample concentrations for these particular studies must be kept pretty low to avoid any polymer interactions that correspond through entangling or physical or chemical aggregation of the polymer. Because physical and chemical aggregation of the polymer traditionally increases the hydrodynamic radius and will in turn make you think that the polymer is much larger than it actually is. One thing to note here is once you avoid these polymer interactions, the diffusion coefficient will decrease linearly with the molecular weight, and that's shown through many different plots. For your traditional polymer species that aren't copolymer blends or polyelectrolytes, the sample concentrations can range as high as 8 weight%, which is more than enough to obtain a very fast proton measurement or even fluorine measurement on any particular species. So in practice, what does this look like? So here I show two plots of PMMA in chloroform as well as polystyrene in chloroform, in which we're varying the molecular weight and determining the diffusion coefficients. We see that they fit linearly. In pulling out the diffusion coefficient, we're able to directly determine the molecular weight. It's important to note here that all of these samples are made at 5 milligrams per mil. And in turn, these strong gradients make these measurements possible with a max measurement time of 15 minutes per measurement. So it's still quite quite fast to measure any of these particular measurements, and we're still not at the limit of our gradient strength. What that means is we can measure even bigger and bigger polymers without having to push the instrument and are still able to obtain very nice data. So in theory, what this application can be used for is for many different things besides very simple polystyrene or PMMA. In theory, polydispersity can also be estimated from these types of measurements. Polydispersity here is basically describing the range of molecular weights you have within your polymer sample. As the range of molecular weights change, so do the plots of the attenuated signals versus gradient strength. You can start to then plot them in a variety of ways to estimate this polydispersity, which corresponds nicely to traditional polymer methods to determine that particular parameter. Also, there's really no limitation to the types of polymers that can be measured. Therefore, you can use copolymers and polyelectrolytes, which are quite hard to understand the molecular weight for through traditional means, to determine the molecular weight using NMR diffusometry measurements. One thing to note here is the sample concentrations will have to be considered, as these particular types of polymers traditionally have much more polymer interactions than, say, your non-charged simple polystyrene or homo block polymers. So what we can do next is we can actually see how NMR diffusometry can be used to determine mixture analysis and to study very complex mixtures. So what I show here is a mixture of pharmaceutical excipients, in which in this particular mixture we have protonated water, which corresponds to that large protonated water signal we see here within the 1D spectrum. We also have a mixture of sucrose and histidine. These traditional signals are also seen within the 1D NMR spectrum. We then have our protein within this excipient, which is this bovine serum albumin. The reason we know that this is the protein signal is just because of how broad it is. It stretches over the course of the entire region, which corresponds to how big and how slow tumbling that polymer or that protein is within solution. We also then have a mixture of ethanol and polymer. This polymer is added by us, it's P188. And one thing to note here is the ethanol actually corresponds from a contamination of our protein. And this contamination is present from the manufacturer. in which using these NMR techniques, traditionally you can deconvolute these signals, but that's typically done through relaxation measurements. And for that to occur, the relaxation of the polymer or the ethanol have to be different enough to where you're going to attenuate one signal without attenuating the other one drastically. If the relaxation is very similar, like it is for this polymer and this ethanol, you're unable to deconvolute these signals, and in turn, another method has to be used. So here we present the use of NMR diffusometry to identify individual components that may overlap within your spectrum that can be used if the diffusion coefficients of these overlapping signals are significantly different. So just a little background here that we haven't touched on. The PGSDE sequence that we're using, or in turn PGSE, will act as a T2 filter process. And what I mean by that is while the gradient is on, all of your relaxation is still occurring within your sample. So what we're doing is while the gradient is actually on, where we're decoding the signal positions, we're removing all of the broad protein signals, as the T2 for that protein signal is now lost before data is actually collected, or before the SID is acquired. And now we're only left with the overlapping signals of the polymer and the ethanol. So how do we know that that is truly ethanol and poloxamer? Well, besides the studies, what we can do is we can plot the neat ethanol in water and the neat poloxamer signal in water and compare that to that excipient mixture. And what we see a nice corresponding to the triplet of ethanol with the doublet of the poloxamer, and you can clearly see that they are quite overlapped within this study. So how are we going to use diffusion to actually deconvolute these signals and to determine how much ethanol is actually present within this mixture? So we're going to go back to the Stokes-Einstein equation, in which if we have different diffusion coefficients for our poloxamer and our ethanol, we can directly determine how much ethanol or poloxamer are in there simply because larger molecules diffuse slower. Our ethanol is a very small molecule, while our poloxamer is quite big because of it being a polymer. And therefore, the hydrodynamic radius of the poloxamer signal should be much larger than the ethanol, and in turn, that diffusion coefficient will be much different than the ethanol's diffusion coefficient. We can integrate these overlapping signals to not only determine the diffusion coefficients of the individual species, being the ethanol and the poloxamer, but also their relative percentage to one another in the mixture. So what I'll do next is I'll go directly back to the instrument, in which this is the actual sample that I loaded into the instrument previously for study. In which the experiment has actually finished running, in which now we see that large water signal. If I were to zoom out, we would see just how much water is actually within this particular spectrum, which is quite a bit. What I'll do is I'll zoom in once again, and now we can start to see some of the sucrose signals, some of the histidine signals, and we see the mixture of ethanol and poloxamer. So what I'll do is I'll zoom in directly onto that region, in which we'll go ahead and integrate this region to pull out the ethanol and poloxamer signals. So what you might say here is, wow, that fit that we're obtaining on the right is a little weird. That's simply because we have two different bi-exponential fits here because the diffusion coefficients are significantly different. As I zoom in to the peak to the right, what we see here is we see the ethanol peaks that are present, but after about the third slice, all of the ethanol has been attenuated out. At that point in time, we see a diffusion coefficient and we see a change in the actual slope that we're obtaining here because now we're just obtaining the poloxamer signal by itself. So what we can do here is we can actually obtain a much longer experiment, start to add more steps of the gradient strength to further identify the ethanol signals from the poloxamer signals and plot them as two separate curves or two separate bi-exponential fits. From those two separate bi-exponential fits, we can directly determine the relative percentage of these. And through things like an external calibration or an internal calibration, we can determine absolute concentrations of the ethanol that have been added as a contaminant from the protein that was within this excipient mixture. So from this, let's jump directly back into the slideshow in which I've already done this analysis. So jumping back into the slideshow, we can see that I've now I ran a much longer experiment, which now I'm plotting the diffusion coefficients or the intensities as a function of our beta parameter, which is everything that we have as a known value within our pulse sequence. And here we're plotting these two different linear fits as two different linear values, in which plotting just the ethanol and the slope from that corresponds to the diffusion coefficient of ethanol, which is about 2.1 times 10 to the minus 9 meters squared per second in this individual sample. while the diffusion coefficient for the poloxamer corresponds to about 1.06 times 10 to the minus 10 meters squared per second. From these fits, we know how much ethanol is in there versus how much poloxamer is from the intensity values that we're integrating. We can then correlate that back to how much intensity we're losing just by applying this individual pulse sequence. And then from there, we can determine how much ethanol and how much poloxamer there are through these relative percentages. So there's about 55% ethanol in here versus 45% poloxamer. And from external or internal calibrations, you can then determine absolute concentrations from this quite easily. So from this particular mixture analysis, now we'll hop into the last topic of this actual webinar, which is the measurement of restricted diffusion to determine structure-property relationships using NMR. So from this, let's bring back that Spitzel-Tanner equation, in which one thing, in order to understand the myriad of material features that drive transport through many different materials, you typically have to reach beyond conventional methods to understand this. And what I mean by that is one method to use that is by mapping out multiple length scales of morphology and pore systems by measuring its diffusion coefficient over those length scales. So NMR, typically by increasing the large delta coefficient, you can start to scan over length scales that range on the order of 50 nanometers up to tens of microns. So if there is a morphological boundary within those length scales, you should see a difference in the diffusion coefficient as the diffusion coefficient should decrease. And what I mean by that is shown here. So let's say we have a particular system in which we have pores that are about 1 micron in length. If we have a molecule that we're looking at for diffusion in this red point and we wait a certain time, we can see that it might run into morphological boundaries if we wait a long enough time, in which these morphological boundaries act as a hard restriction, which are going to stop the diffusion coefficient, and in turn, the diffusing species is going to bounce off of that and then basically go back to where it began, more or less. In turn, you now see a slowing of the diffusion coefficient as it's been blocked by those morphological restrictions. So if you then plot the diffusion coefficient that's obtained as a function of the diffusion time, you can now start to assess the material heterogeneity that will directly affect transport. And how this is done in practice, let's say we have two different membranes. So these are two different porous membranes of PEO that change in the weight percent of PEO, in which shown in blue is a much larger PEO weight percent of about PEO 30, because 30 weight percent being the PEO component of this copolymer map, while the red is going to correspond to the PEO at a 10 weight percent. What we see is the PEO 30 has much larger pores, and in turn, the diffusion coefficient does not change as a function of the diffusion time. But for PEO 10, the pores are much smaller, in which as we start to probe the diffusion coefficient as a function of the diffusion time, we see a decrease in that diffusion coefficient. That decrease in the diffusion coefficient, once it starts occurring, can then be tracked back with the root mean squared displacement equation to determine the length scale of when those boundaries occur, in which here the boundaries start to occur at around 3 microns for this PEO membrane. So this can be done with many semi-solid materials and is quite readily done for reverse osmosis membranes, as that is one of the main functions of these reverse osmosis membranes to block things like lithium or other hard metals from going through and contaminating the water. So overall, we've showed a myriad of things. We've discussed the use of the Spinsolve to measure NMR diffusometry in many different applications. These certainly are not the only applications in which NMR diffusometry could be used, as it is quite a powerful technique that can be used to determine many different things. Using the Spinsolve and its interface to measure these particular measurements, we hope to limit the amount of technical expertise and know-how that is needed to actually perform and get accurate data from these measurements. So thank you for your time and spending your time with us today. If you have any questions about the webinar, please type them into the questions box, and we will try to answer them shortly in our Q&A session that's to follow. Please do not hesitate to contact us with any questions, and make sure to visit our homepage for the latest updates in terms of development of the Spinsolve and its various applications. Last but not least, I'd like to give a few more minutes for questions to filter in, and I hope you all enjoyed the webinar, and please stay tuned for our upcoming webinars on many more exciting applications and uses for the Spinsolve, such as automated sample identification through our QID Solve software,